Very good morning to one and all. My name is A. Indumati, Assistant Professor of Zoology Department. Today we are discuss about the topic denaturation and renaturation of DNA. We all know that the DNA is a double stranded molecules. It is stabilized by chemical interactions. Most important interaction is hydrogen bonds. Also, we know that adenine also paired with thymine, where two hydrogen bonds, and cytosine also pairs with guanine, where three hydrogen bonds. Inside living cells, the double strand of DNA separate from each other during DNA replication. This separation disrupts the hydrogen bonds between the bases. And this task is done by a protein is called as helicases. In a laboratory, hydrogen bond of DNA double helix can be disrupted by two methods. First method is by changing pH of the DNA solution, and the second method is heating the DNA solution. Let's talk about the DNA solution is heated as a result, hydrogen bond disrupted, and double stranded DNA is separate into single strand DNA. This separation of DNA strand is known as the denaturation or melting. Thus, denaturation of DNA is the loss of helical structure of the DNA as the temperature is increases. This percentage of DNA denaturation is also increases. This can be shown graphically, and the curve obtained as the melting curve of DNA. How to measure the DNA denaturation? DNA denaturation measured by using spectrophotometer. This is based on the fact that all nucleotide bases consist of aromatic rings. These aromatic rings absorb light in the ultraviolet range. Now, nucleic acids such as DNA are made up of with these bases. Therefore, DNA molecules are also absorb the light of UV range. All the bases of DNA like adenine, guanine, cytosine, thymine have strong absorbance of at 260 nanometer. Double strand of DNA absorbs less light at 260 nanometer compared to single stranded DNA. This is because base stacking interactions in DNA double helix interference with the absorbance. So as the DNA denature of melt, its absorbance are increases. Absorbance of DNA solution was naturally under particular set of conditions. And the sigmoid shape curve obtained the plating the absorbance bar used of increasing the temperature, perhaps in the melting curve of DNA. Now, the lower portion of the melting curve represents that all the DNA molecules in the given DNA solution of interact. That is their double stranded, and this upper portion of the melting curve represents all the DNA molecules have completely denatured or melted. So here we can say that as temperature of DNA increases, DNA molecules melt and the two strands are separated. Also, at DNA becomes single stranded. Its absorbance is increases. The temperature at which half of the DNA molecules are denatured is called the melting temperature of DNA. That means at this temperature of half of the DNA molecule present in the solution will be the single strand and other half will be the double strand. Melting temperature found at the midpoint of the melt. This things kept besides the denaturation. Another feature of DNA about helix at separate the complementary strand of DNA can spontaneously reassociated to form a double stranded. This happens when a temperature of DNA solution is lowered below melting temperature. This phenomenon is known as the DNA renaturation or annealing. At this end of this video, we understand that DNA melt and reanneal reversibly. Thank you.